Hey everyone! So I figured I'd bring you into my basement classroom for now. There's you. And do a little review of how gene expression is controlled in bacteria, and that's the operons. So we're looking at just lac versus trip. So let's go over a couple of different things. All right, so I'm going to bring back some terminology first. When we look at the metabolic pathway, is it going to be anabolic, meaning that it's building something, or is it going to be catabolic? Is it breaking something down? Okay, so if we look at the lac operon, right, the goal of the lac operon is to be able to create the enzymes that actually break down lactose. So in this case, this is going to be a catabolic metabolic pathway. And the trip operon in prokaryotes is used to make tryptophan, an amino acid, when there's not enough in the environment. So this is going to be an anabolic. Let me get my head out of the way so you can see it. All right, so operons can either be inducible or repressible. Well, what the heck does that mean? Well, to induce means to turn something on, that's normally off, and to repress something means to shut it off when it's normally on. Okay, so if we look at the lac operon is normally off, so that means it's going to be inducible. Okay, so over here we're going to put normally off. It is inducible. And the reason for that is that normally the prokaryotes, their normal food is glucose. So as long as there's enough glucose in the environment, then it's not necessary to break down lactose, which is a disaccharide, into glucose and galactose. Okay, so normally, as long as there's enough glucose, the lac operon is off. Trip is the opposite of that. Okay, so trip stands for tryptophan, the amino acid. So this is normally on. The bacterial cell is creating its own tryptophan. When there's enough tryptophan in the environment, though, it will be repressed. Okay, so this is called a repressible operon. That's the difference between the two. All right. So if we look at how this works as inducible or repressible, then we can say, okay, is the repressor that's created by the regulatory gene being transcribed into an active or inactive repressor, is the repressor going to be active or inactive? Well, since the lac operon is normally off, then in order to block RNA polymerase, this is an active repressor that's sitting inside the operator, okay? So this all, I want you to remember, this goes back to protein synthesis because that's what's happening here. And an operon is a sequence of genes. Starts off with a regulatory gene that is transcribed into either an active or an inactive repressor, in the case of TRIP. And then upstream of that, if you go back and you look at the diagrams, you will have the operator, and the operator is where the repressor will actually bind to. So if it's active, if that repressor is sitting in the operator, then RNA polymerase cannot get past it to transcribe the genes to create the enzymes, in this case, to break down lactose. However, if it's inactive, then it means it's not blocked, and RNA polymerase is transcribing the genes to help create the enzymes that will be important in the making of tryptophan. Okay, so that's the difference between the two. All right, so in this case, what is the regulatory gene? The regulatory gene controls for the transcription of the active repressor. And for tryptophan, it's going to control for the transcription of an inactive repressor. Okay? All right. So, what changes in the environment to actually turn these genes on or off? Okay? And that's what we mean by gene expression. All right. So, in the environment, if we look at, we'll add environment down here. The lac operon will turn on when there's not enough glucose. Okay, so in that case, what happens is lactose, actually it's like a form, it's like allolactose. Allolactose will actually come in and it will bind with the active repressor, and that causes a conformational change, and it pulls it out of the operator, and then RNA polymerase will transcribe the genes to make the enzymes to break down lactose. All right, so if there's not enough glucose in the environment, that's when it becomes inducible and it will turn on. Over here, if there's a threshold, if there's enough tryptophan that it's not necessary for the bacterial cell to continue making it, because that would be a waste of energy, then the tryptophan will actually bind with the inactive repressor, and then it activates that repressor, which will go sit in the operator, and it will shut off and repress the trip operon that's normally on. Okay, so that's how they work. All right, now the only other difference between the two that I taught in the video lesson was that with the lac operon, there can be some upregulation, right? So there's a form of cell communication and positive feedback over here when there's not enough glucose in the environment, and that will activate the production of cyclic AMP, which sort of gives like a boot in the butt to RNA polymerase, so that it's actively transcribing the genes to make lactose in a much faster way. Okay, so if you don't remember what that is, go back to the video lesson and take a look at that upregulation again. That would be a form of positive feedback because it would be a signal for this to increase in speed, right, once we turn it on. Okay, so that's the basic difference between the lac and the trip operons for prokaryotes. So if you have any other questions on that, definitely shoot me an email or send me a question through Remind. Okay? All right, so I think a good thing to do would be to take this and then compare it to eukaryotes. So I'm going to erase our little T-chart now. And we'll put up a different question here. Let's erase this. And the question we'll ask now is, how is gene expression controlled in eukaryotes? And remember, eukaryotes do not have operons. Okay, that's only for prokaryotes. All right, so eukaryotes, because everything is decoupled, meaning that inside the cell things are compartmentalized. You have a nucleus, and then you have cytoplasm, where translation takes place. There's lots of different control points for eukaryotic gene expression. I think the easiest ones to remember are to go back to protein synthesis and talk about what happens to the pre-mRNA inside the nucleus. I think that's a very easy thing to do, okay? We can also look at the structure of DNA itself and how it's wrapped around special proteins called histone proteins. We can look at the methylation or the acetylation of the DNA, which is involved in opening that up. And we can look at some post-transcriptional stuff, okay? So for a very quick recap, if you're inside the nucleus, right, and just remember, things are compartmentalized. right? It means there's the nucleus surrounded by a nuclear membrane and cytoplasm. And so this gives lots of control points in regulating gene expression in eukaryotes. Okay, so if we look at the first one, we can say access to DNA, right? So if you recall, DNA is tightly wound inside the nucleus. There's a lot of it, so it has to be tightly wound. And it's wrapped. Histone proteins wrap it up and coil it up. So one thing that can happen is acetylation, also methylation, the adding of methyl groups, which can either unwind or wind back up the DNA. Okay, so acetylation means acetyl groups are added, and it takes the histone proteins and it unravels them, and then that makes it available for a section of DNA to be transcribed or not, right? So that's a control point. And then we can look at things like how RNA is processed. Which we learned all about with protein synthesis, right? And so this is the 5' cap, the poly A tail, right? So those have to be there in order for the transcript to just even make it out of the nucleus so that it can be expressed and translated into a protein, right? So that's a control point there, number one, right? And then also with this RNA processing, you can have alternative RNA splicing, right? So remember that exons exit, right? So introns are cut out. So the introns are still cut out. That still happens. But what can happen is that you can splice out some different exons, right? So let's kind of do a little drawing of that. So let's say we've got, I'll do line here. Here's your transcript, okay? And I'm going to divide that up, and we'll number it out like this, okay? And I'll call it, we'll just give it a number. So this will be for gene 1, this is 2, 3, 4, and 5. And so those all represent the exons. And of course, you've got your 5' cap, you've got your poly A tail. Okay, so we can control how this is expressed and how proteins are produced by also alternatively splicing out some of the exons. Okay, so like let's say for a particular need for a cell, we splice out 2 and 3, and so that means for our transcript, right, obviously it's going to be shorter, and so now we've got 1, 4, and 5. Right? And so if that's what's going out into the cytoplasm to get transcribed into a protein, you can see how that's going to control the expression of the genes, right? And expression means, you know, what's the phenotype? What physical thing might we see in a trait, or what protein's being produced from that gene? Okay? So that's what alternative RNA splicing is. You have an infinite number of combinations in eukaryotes. That's something you're not going to see in prokaryotes. All right? So we've got access to DNA, we've got the RNA processing. I think these are the easiest ones to kind of remember. You've got your alternative RNA splicing, that's here. And then once you're out into the cytoplasm, you know, there are other things, or even on the way out, right? So like, got to get past RNAi, or RNA interference that would actually chop up the transcript, so that's also another way of regulating gene expression, because if the transcript doesn't make it out of the nucleus, then it's not going to be expressed. Okay, and then once you're out there, you can have post-translational control. . right? And in the video lesson, I was talking about ubiquitin, right? So it's a special protein that's ubiquitous throughout the cell. So in combination with peroxisomes, they can actually destroy some of the proteins that are produced there, and that will also regulate how gene expression is controlled. Okay, so just a simple review of some of the more important things for control and expression in eukaryotes. So hopefully that's helpful to you, and then it wraps some things up, and I'll see you soon.